Thank you, Dr. Kaur, for this kind introduction, and I'm happy to be part of this first international conference on data science in biology. And I also thank Abhishek for the kind invitation to this conference. So when I listened to this previous talk, so I noticed that my talk will be much more translational than the at least most talks in this conference, but I hope that you will be able to follow me. So I will be talking today about germline genetics of cancer, and I will show you a couple of examples of our own studies on this field. And before coming to that, I would like to introduce you a few terms on this germline genetics. So the first one is heritability, so which means or indicates how much of the cancer risk is explained by inherited genetic factors. And twin studies have been a valuable tool to evaluate heritability and also environmental components of cancer. And as you can see from this table, so the common cancers, prostate, breast, colorectal cancer, so they have a heritability something like 30–40%. If we then look at the proportion of cancer, which is familial cancer, and here by familial cancer I mean that in the family there are at least 2 cases, two first-degree individuals who have the same cancer. And the proportion of familial cancer of all cancer in prostate cancer is something like 20%. In breast and colorectal cancer, it is less than 15%. Then what we are knowing about the genes behind this familial cancer. So the proportion of familial cancer explained by the known gene is highest in breast cancer, where something like 50% of the genes are known, or the families get an information which gene could be the reason for their cancer. Then in prostate and colorectal cancer, so the proportion is around 30%. What are then these genes which are known to affect familial cancer risk? So here in the scale of variant allele frequency in the population and cancer risk size, so we have at one end the rare high penetrance cancer genes, which were identified mainly using lineage analysis in families. And here you can see the well-known breast cancer genes BRCA1 and BRCA2, and then also the known colorectal cancer genes APC and mismatch repair genes. At the other end of the scale, we have a large number of common low penetrance alleles, which have been identified by genome-wide association studies. Individually, these variants cause a very low risk, but then in combination, they can have a substantial effect on cancer risk. And between these two loci, we have very few moderate risk alleles. And what we are doing now, so we are using the next generation sequencing techniques, whole genome and whole exome sequences to identify more high penetrance or moderate risk alleles. And we are also contributing to the identification of the low penetrance alleles, common variants, using the genome-wide association study approach. And then I start with the first example, and here we have coming back to the family-based studies. So we are trying to identify families with many cancer cases, and we are trying to also collect blood samples from as many cancer cases as possible, and then also from unaffected healthy individuals in each family. And we hope also to recruit some individuals from the older generation of the family. So by that, we try to guarantee that a single gene defect is the cause of the disease in each family. This is then the pipeline which we have developed in our division. And Abhishek Kumar has been a key person in developing this bioinformatics pipeline for this project. So we start here with identification of families together with our collaborators who have collected a large number of familial cases and families. And then we collect the DNA sample and isolate or blood sample and isolate the DNA, or our collaborators are often doing that also. When we then get the DNA sample, we submit it to our core facility at German Cancer Research Center, and they are doing the whole genome or whole exome sequencing. They are doing the variant calling and variant annotation. And here our main scientific collaborators have been Matthias Lesner and Nakarayan Paramasivam. After that, we get all the variants with variant allele frequency less than 0.1% to our lab, and we submit them to the pedigree-based variant segregation analysis. And only the variants which are segregating with the disease in the family, so they are then going further to this bioinformatics part of the pipeline. And here we classify the variants based on their location in the genome. We start with coding variants, which are the most likely to be the functional variants in the genome. And we used something like 10 tools to evaluate the deleteriousness of the variants. Then we used some 5 tools to evaluate the intolerance of the corresponding genes against the functional variants. And we used also 3 tools to evaluate the conservation of the variant position itself. Then for the non-coding parts, we have also divide that also in different parts, like 5' UTR, 3' UTR, intergenic variants, and we use different bioinformatics tools to evaluate their influence of gene regulation. After that, we have something like 10-20 variants, which we then submit to a thorough literature search. And after that, we may have something like 5-6 variants, which we then go for further with validating in additional families. And many times this is done by our collaborators and sometimes by ourselves. At the same time, we are also doing functional evaluation in our lab, and there Apul Pandapalli has been a key person behind these assays. And then Manda, Peping, Lisen, Sara, and Ayushi have been the persons working in the lab on this project. Okay, but here is then anyway, so that the families which we have sequenced so far, or now we have maybe somehow something like 300 families, or 300 genomes sequenced, whole genome or whole exome sequenced. The largest number of these families are colorectal cancer families, but we have also a large collection of multiple myeloma families, and then we have some pancreatic cancer, thyroid cancer, Hodgkin's lymphoma, cervical cancer, and neuroendocrine tumors as well, which we have sequenced. And here I give the examples on the colorectal cancer family analysis, and here you see the 4 families where we identified mismatch gene mutation. So you can see here in these pedigrees that we have colorectal cancer cases which are indicated either with the black or red circles and squares, and so that we have colorectal cancer cases in 3 generations in most of the families. And the variants which we identified in these families was a brain shift mutation which caused a stop codon. Then we had a splice site variant which also caused a brain shift, and then immature protein translation. And then we had one large exonic duplication in one family, disturbing also the protein. And then in one case we had a missense mutation which was predicted to be deleterious. Then as an example how our pipeline is working in our families, so I take up this family where we have 3 colorectal cancer cases indicated by red, and 2 polyp cases in the family. So these individuals have colorectal polyps and maybe an increased risk of getting colorectal cancer. Then we could also sequence one healthy individual who was unfortunately very young, 23 years old. And in this family, we identified a protein tyrosine kinase 7 missense variant. Here, here you can see also how our pipeline is working with the decreasing the number of variants. So this family was whole exome sequenced, and we identified altogether 10,500 variants with allele frequency less than 0.1%. 421 of them segregated with the disease in the family, 72 were exonic variants, and 47 non-synonymous variants. Then we used a general deleteriousness prediction score, a kind of meta tool, to identify 32 variants, which were among the top 10% of the deleterious variants in the human genome. 23 of these variants were located in the conserved. 14 were located in the genes which were intolerant against functional mutation, and after we used the 10 deleteriousness tools, we ended up to 6 variants. And these variants you can see in this table, and we focused on to this protein tyrosine kinase variant, because it is involved in Wnt signaling pathway, which is an important pathway in colorectal cancer, and it is involved in cell adhesion, migration, polarity, proliferation, and so on. So here is our original family again, and we confirmed the segregation in this family by using Sanger sequencing. So the variant was present in these 3 colorectal cancer cases, and it was present in one of the polyp cases. It was not present in this other polyp case and not in the healthy young individual. But then we screened this variant in an additional 1,700 familial colorectal cancer cases, and we identified another individual who carried this mutation, and the father of this individual and one of his half siblings also carried this mutation. So now we have 2 families where the same mutation was identified. This variant is located in the extracellular domain of the gene, so it is located in one of these IG loops, and there are some publications showing that the canonical Wnt ligands like Wnt3a bind to this extracellular domain of the PTK7 and initiate the degradation of the protein. So we hypothesized that this binding may be interfered by this variant and inhibit PTK7 degradation. Then we tested if this protein is more stable in the when it is carrying this mutation, and we introduced this mutation into the colorectal adenocarcinoma cell line HD29, and indeed, so we observed that there was an enhanced PTK7 protein expression compared to in the wild type cell, in the mutated cells than in the wild type cells. We also observed that there was an increased cell proliferation in the mutated cells compared to the wild type cells, which are shown here, the mutated cell proliferation in the red line and the wild type in the blue line. Then we went on to look at the downstream targets of PTK7, and we observed that in the variant cells, the expression of the downstream targets of PTK7, CREP and P53 were decreased in the RNA level, and we also confirmed that in the protein level, as you can see here, for CREP and PTK7 in the mutated cells and then in the wild type cells. Then we also noticed that there was an increased phosphorylation of AKT in the mutated cells compared to the wild type cells. And that indicated that this mutation may interfere in the apoptosis or activation of the AKT signaling. Sorry for that, so I don't know why it is. Yeah. Okay, so as conclusion for this part, so we have developed a familial cancer variant prioritization pipeline that can prioritize both coding and non-coding single nucleotide variants, small insertion deletions, and copy number variants in the whole genome sequencing data. And this data can be used then for further functional validation. In managing cancer risk, so it is important to recognize risk factors, identify high-risk patients, and implement appropriate surveillance. And we have contributed to this in this study by identifying a novel missense mutation in a protein tyrosine kinase PTK7 that potentially inhibits apoptosis pathways by downregulation of its downstream targets CREB and p53, and which activates AKT signaling by increasing AKT phosphorylation. As clinical importance, our study may help in genetic counseling and personalized therapy of the families. And it may also help in development and implementation of individualized treatment options. Then I move to the second part of my talk, where I explain or show you one example of our approaches on, or our studies on using whole genome association study approach. So here we collect a large number of cancer patients and a large number of healthy controls from the same population. Then we do the genotyping using the Illumina single nucleotide polymorphism sheets, and then we do quality control using both sample-based quality control and SNP-based quality control. And here we exclude all the samples where the SNP call rate is low, where the individuals are not European ancestry, and from related individuals we only take one into our analysis. And in the SNP-based analysis also, so we exclude all variants with low call rate, with low minor allele frequency, and where the genotype distribution is not according to Hardy-Weinberg equilibrium. Then when we have this high quality data, so we impute all the variants which are not directly genotyped in these Illumina sheets. Then we do the association analysis, and here we use the genome-wide significance level p-value 5 × 10⁻⁸, and suggestive levels of significance p-value 1 × 10⁻⁵. Then in most cases, so we have either replication population for the best associations, or in many times also the whole genome association study from another population, and we pool these in the meta-analysis. Then we can also use the stratified analysis based on the subtypes of the cancer. And at the end, we of course have the most important part to interpret the results using both publicly available data and then also functional testing. So here are the situation of the known risk alleles, of the known common risk alleles in cancer which have been identified by genome-wide association studies. And those ones which are underlined here, so that are the cancer types where we have contributed. And I will take up the myeloma as an example, because there we have basically identified all the so far known common low penetrance alleles. So multiple myeloma is a plasma cell disease which originates from the post-germinal center B cells. It goes first to the precursor stage, monoclonal gammopathy of undetermined significance. Then it can stay in this stage, but then it can also develop further to multiple myeloma or AL amyloidosis. Multiple myeloma can then, in rare cases, also lead to plasma cell leukemia or AL amyloidosis. Here are then the diagnostic criteria of these different stages of the disease, and it is based on the M protein level. So this is an immunoglobulin which it is, they are expressing then on the bone marrow plasma cell count and end organ damage. And here you can see that AL amyloidosis and multiple myeloma are the only ones where there is an end organ damage. For multiple myeloma, there is also a CRAP criteria, which means that calcium is elevated, there is a renal failure, anemia, and bone lesions. Then there are also a lot of genetic events during the course of the disease. So the primary genetic events involve usually the immunoglobulin heavy chain translocation together with the known oncogene like cyclin D1 or fibroblast growth factor receptor 3. Or then there are also multiple myelomas with hyperdiploidy, and there are trisomies in the odd chromosomes or in the most odd chromosomes. Sometimes there is also a combination of both of these. Then there are also secondary genetic events which are often chromosomal regional deletions or gains. There are also some mutational events, most commonly NRAS, KRAS, and BRAF, and then also some new translocation, usually other translocations not involving the IGH. Then I will show you the results of our latest meta-analysis, which was published in 2018. So there we had some 8,000 cases and 240,000 controls with three genotypes from the UK, Germany, Iceland, USA, Norway, and Sweden, and the Netherlands. And we replicated this study in 1,700 cases and 6,000 controls from Germany, Denmark, and Sweden. Here are then the results in the Manhattan plot scale. So here on the x-axis, you can see the position of each SNP on the genome according to the chromosomes, and in the y-axis is the significance of the association as minus logarithm p value. And this red line here shows the genome-wide significance level, and the blue line the suggestive significance level. And I will show you later on an example of the importance of this most robust locus in this analysis. So in multiple myeloma, there are no high-risk genes known, and these 23 loci are the ones which have so far been identified as low penetrance risk alleles, and we have estimated that something like 60% of the heritable risk is attributable to all common variation in the genome. And because most of these variants identified only here in this study, so are located in the non-coding region, so there is it is important to functionally evaluate the reason for this association. So that we are doing using both publicly available data and then also doing by ourselves some experimental analysis. So we can evaluate the chromatin state of the associated SNP or linked SNP, so that like if it is located in active promoter or active enhancer, We have expression data available for the malignant plasma cells, so we can look at their EQTL effect, so that if they are affecting the expression of the nearby genes, then we can also look for if these variants are enriched in the locations where there are active histone marks and the histone is available for expression. And then we can also do promoter capture high-C analysis for long distance chromatin interactions, so that if the SNP locus is interacting with the promoter of a target gene. Then I come to the functional relevance of the C7p15.3 locus. So this variant is located in this DNA H11 gene, but it maps to the IRF4 binding site, and IRF4 is an interferon regulatory factor 4, which is also known as myeloma oncogene 1. It is located in the strong enhancer element, and it is a strong EQTL for the nearby or neighboring gene CCDCA7L. This gene encodes a MYC interacting protein, and it is an important partner for this pre-B cell differentiation and B cell development, and MYC is of course an important gene in cancer development as well. So when we knock down this IRF4, so we notice that in addition to suppressing the expression of IRF4, also this target gene CCDCA7L was suppressed, and this suppression led to the decreased cell proliferation and induced apoptosis. Then we have also done stratified analysis using the cytogenetic data, which is available from Germany and UK for something like 1700 cases. And here we identified cyclin D1 splice site polymorphism as a risk factor for T1114 multiple myeloma. And this is the first time when a constitutive genetic factor was associated with the risk of a specific chromosomal translocation. This locus is very interesting in that sense, so that it has two transcripts. So the transcript A, where there is the G allele on the splicing site, is the full-length cyclin D1, and A allele causes an alternative splicing, and it leads to an alternative transcript. And it is interesting that the G allele is the risk allele for T1114, but then the A allele is the risk allele for cancer. In this B transcript, so we can conclude that it has a greater transforming capacity, but it lacks a DNA damage response part, which is associated for cyclin D1. So we hypothesized here that the G allele may facilitate clonal expansion after the translocation event and overexpression of cyclin D1. G allele may also affect the early evolution of T1114 multiple myeloma, because we observed a similar association in the precursor state of this disease. And most importantly, G allele is a major risk factor for AL amyloidosis, where T1114 is much more common than in multiple myeloma. So in AL amyloidosis, the frequency of T1114 translocation is over 50%, and in multiple myeloma it is about 20%. So that is also confirms the function of this variant in the T1114 type of myeloma and AL amyloidosis. So in this part, I have shown you an example how a germline variant may affect the MYC pathway, and I saw also another example how the germline variant affects cell cycle regulations. Other SNPs which we identified to be associated with multiple myeloma, so include autophagy and plasma cell biology, telomere biology, chromatin remodeling, and immunoglobulin production. So here are our collaborators for the colorectal cancer GWAS whole genome sequencing project. Professor Jan Lubinski from Poland is our main collaborator, so he has a large collection of familial cancer cases of 4 different cancers. Then in the multiple myeloma GWAS project, Hartmut Goldschmidt, Richard Holston, Martin Kaiser, Björn Nilsson, Karet J. Morgan, and Kari Stefansson and Peter Sonnefeld have been our main collaborators. Per Hoffman is an expert on this GWAS analysis and our long-time collaborator. Then the whole genome sequencing and data management was done in the core facility of our institute. Then Abhishek, Opul, Peping, Manda, Ayushi, and Lisen in our group were involved in this whole genome sequencing part and identification of the high-risk variants. Hauke, Mikuel, Povang, Supayan, and Kiara have been participating in this myeloma project. Kari Hemminki was the head of our division of molecular genetic epidemiology, which was closed last summer, and now he is a professor at Charles University in the Czech Republic. Then in the bioinformatics part of our work, so as I told already, Matthias and Anaka have been our cooperation partners. So with that, I thank you, and I'm happy to take some questions.